ஹாய் ஹலோ வணக்கம் வந்தம் நமஸ்தே நமஸ்காரம். நான் உங்கள் நண்பன் விக்கி ரைட். யூஆர் வாட்சிங் விக்கி யூடியூப் சேனல். So in the video in this video what I'm going to tell you is I'm just going to talk about the RAPD. So many of you may hear about the term called as RAPD, and many of you may not know about RAPD. RAPD is nothing but it is expanded as randomly amplified polymorphic DNA. It is simply called as RAPD. In other terms, it is referred as randomly amplified polymorphic DNA. So we already know what is DNA and other kind of things. Those who want to work in the molecular diagnostic, or in order to work on the molecular markers, people will be targeting the first prioritized marker is RAPD. So RAPD is a kind of marker where you can able to detect some kind of bands that will be useful for your own studies. It depends on your own studies, and the result will be interpreted based on the study that they do. So here I'm just going to simply do an experiment to perform RAPD and try to know more about RAPD. So many of you may hear about the RAPD, SCARE, RFLP and many such kind of markers, but people always used to confuse between RAPD, RFLP and AFP. By this video you could able to really distinguish what is RAPD is. So RAPD is a randomly amplified polymorphic DNA. It means you have to amplify the DNA at random parts. So DNA is it's like said to be like it is a very large biomolecule that has been present in our cells. Not only our cells, it is present in all over the biological materials. So the DNA which is there inside a cell will be amplified in a random spot. So random spot means we will not focus on some particular parts. It will be amplified in a random spots, and the randomly amplified DNA will be run through gel electrophoresis, where we could able to find the difference among the group, or difference among the population, or else the similarity among the population using the band that has been present in the gel. So here I'm just going to perform RAPD. It is a very simple protocol to distinguish between the bacteria. So here we used RAPD for distinguish between bacteria. So we'll be isolating bacteria from some source, or from some kind of element, or some kind of sample that we employ, but we'll be getting many more bacteria. In order to distinguish between the bacteria, whether we got the bacteria morphologically may be similar or morphologically may be different, but genetically whether it is similar or different, we have to identify it. So we have to identify it. In order to identify it, we are performing RAPD. So we are employing the marker called as M13 marker. It is a universal marker, we can purchase it anywhere. So M13 marker will amplify in some particular part of the bacteria. So based on that random amplification, we could be able to figure out the differences among the population that we have isolated, or the similarity that we could able to find out among the population. So I am just going to employ 17 bacteria and I'm going to make RAPD. So in this single video you could be able to find a procedure for RAPD, hands-on experiments on RAPD, as well as you could be able to find the result that I'm going to share with you. So in order to make RAPD, you should have 5 different things. So first of all, you should have a PCR mixture. So it will be having tag polymerase, it will be having DNTTPs, and it will be having some magnesium and everything which will accelerate the PCR reaction. So I have the ready mix PCR amplifying mixture, so which could be able to amplify the particular DNA with its own primer template. So I'm having the tag polymerase added PCR master mix, and I'm having the DNA with me, the bacterial DNA, the bacteria which I want to distinguish or which I want to find the similarity. So I am having the bacteria, that bacteria DNA is with me. Then I'll be adding M13 primer, water and the PCR. So we should have all these things together so that we could make RAPD. We can perform RAPD. And one more information, for the bacteria DNA isolation, I have uploaded the previous video, so you can able to get a protocol from that video. So that video dealing with the very short DNA isolation protocol, where you can spend just 10 minutes and the preparation time just 2 to 5 minutes. So within 15 minutes you can able to isolate the bacterial DNA. So I have already isolated the bacterial DNA from the last video. So this is the sample. The person who's already watched the last video can able to distinguish this one. So this is the 17 bacterial isolates DNA. So I have isolated DNA from the 17 bacteria, and now I'm going to perform RAPD using M13 primer. So this is the master mix that we have already purchased. So I'm going to use this master mix for this RAPD, and then M13 primer. So here it is. So I have M13 primer. So this is the M13 primer, and this is the working stock of the M13 primer. So I have already prepared a working stock. So working stock of the primer, M13 primer is here, master mix is here, and autoclave water, which is deionized water. So I'm going to use this one. So the water is here, master mix is here, and the M13 primer is here, DNA template is here. So I'm going to use the 17 bacterial DNA for this study. So I have already made a calculation. I'm going to do a 20 microliter reaction. So you can fix a reaction based on your own convenience. So people normally will be doing 50 microliter or 25 microliter. So I'm here going to do a 20 microliter reaction. In a 20 microliter reaction, I'll be employing nearly 10 microliter of the master mix. So 10 microliter of the master mix, 2 microliter of the M13 primer, RAPD primer. This is called as only the forward reaction, forward primer. So for your case, I could able to tell you like for the 16S sequencing or for any kind of sequencing, we will be adding forward primer as well as the reverse primer. But in order to perform RAPD, you can employ only one primer, whether you can employ forward primer or you can only employ reverse primer. So I am employing M13 forward primer alone, so which is enough to make our randomly amplified polymorphic DNA. So RAPD, for RAPD I am having M13 forward primer and 17 bacterial DNA. So I will be adding 10 microliter of the master mix, 2 microliter of the RAPD primer, 2 microliter of the each bacterial template in each wells, and nearly 6 microliter of ionized water. So I'm just going to make the mixture for all the 17. So later I could be able to add mixture from this tube to all the each other tubes. So for making 17, I have already made a calculation for 17 bacterial DNAs. I am going to prepare the master mix and I am going to add it. So for the 17, I'll be adding 10 microliter each. So I'll be adding 170 microliter of the master mix to this one. So all the things are pre-autoclaved. So I'm using the pre-autoclaved things here. So 170 microliter of the master mix. I have already made a freeze star and everything, so I'm directly adding. If you're doing it by right now hands, so you have to freeze those and you're doing it. Okay, after 170 microliter of PCR master mix, I'm going to add water. So water is almost for 17 sample. I'll be adding 60 microliter of water for the each sample. So totally 17 into 6, you will be having 104 microliter of water. So I have added water. Then what I need to add M13 primer. So for M13 primer, I will be adding 2 microliter for each sample, each bacterial sample. So totally 17. I will be adding 34 microliter of primer into this oil. So here it is M13. So now it is having the master mix, the water and the primer. So I can directly add this to all the tubes so that I could be able to add DNA on the respective tubes. So before that I would like to spin it a little bit. So I have taken 17 PCR tubes and 17 PCR tubes cover caps. So I have taken 20 microliter of the pipette. So I'm going to add 18 microliter of this PCR mixture to all of this. So if any of you want to have a look at on the DNA isolation protocol, I have given the video link in the description, so you can find the first video from the link. So after this, we are going to add 2 microliter of the DNA on each tubes. So this is the DNA that I have already isolated from the video number 1. So if you wish, you can able to go back to the video 1, you can watch it and you can come back. நான் தைவானில் இருக்கிறது எனக்கு சந்தோஷமாக இல்லை, ஆனால் பீசனுக்கு நல்லா சந்தோஷமாக இருக்கும் போல். தைவான் வந்து 100 நாளில் எனக்கு ஒவ்வொரு நாளும் விஷ் பண்ண ஒரே ஆள் பீசன் மட்டும். ஹாப்பி ஸ்டே இன் தைவான், பெஸ்ட் விஷஸ் ஃப்ரம் பிஎஸ்என் மெசேஜ் அஞ்சு. தேங்க்யூ பிஎஸ்என்எல். By the way, how to come to Taiwan for the TEEP internship and how to live in Taiwan and what are the precautions that you have to take before reaching Taiwan and what are the cell phone network that you can use in Taiwan, everything has been uploaded in the travel abroad Taiwan playlist as well as the TEEP playlist. If you want, you can check that also. So I have said 2 microliter, so I'm just going to take DNA from each tubes and I'm going to apply it to all the each tubes. So I have taken the samples from 1 to 8 and I'm going to add from 1 to 8 of these tubes. So while adding the DNA to the tube, you have to add it in the periphery so that it will get in. You have to be assured that you are adding in a right tube. பேய் கூட நேரத்தில் போய் ரெஸ்ட் தூங்கிட்டு இருக்கோம். விடிய காலில் 5:30 மணி உட்காந்து டிஎன்ஏ ஐசோலேஷன் ஆர்ஏபிடி யூடியூப் வீடியோ நல்லா வருடா இருக்குங்க சார். So first 8 samples has been added. I'm going to add the rest of the samples. Sample number 9 to 16. If any of you wants to do internship in abroad, especially in Taiwan, you can check my video in the playlist called as TEEP, Taiwan Education Exchange Program, where you could be able to apply to the various professors and the various institutes in Taiwan. If you've been selected, you can come here to do your 6 months internship in Taiwan with a fellowship of 15,000 new Taiwan dollars. If you wish, you can come. If you wish, you can apply. If you got selected, you can come. And I am also giving that video link in the description. You can check it out. So I have added 16 samples now. One more left. Last one. Okay, now I have added a PCR template, DNA template, PCR amplicon, master mix, and with the water. So it is ready. Now I want to do only one step, that I have to little bit spin it just to bring all the things together. So here is the PCR machine. So I'm going to start the PCR machine. I am going to do RAPD. So I am using M30 ml. So this is the default program, the RAPD set. You can put 95 degrees Celsius for 5 minutes as a step 1 initial denaturation. Then the 90 for 1 minute denaturation. 40 degree for 30 seconds annealing. 72 degrees Celsius for 3 minutes 30 second extension. And 72 degrees Celsius for 6 minutes final extension. And this is 4 degrees Celsius overnight in case if you want to go for it. So I have opened this one and I'm gonna place my sample. So these are my sample, 20 microliter sample I have placed. Just to make sure that it is being fixed well. So I'm closing this one, and this temperature is already set, so I'm just going to run it. The sample is 20 microliter. I have pressed it and let's start. So after the pressure running off, I'll be just taking the samples and I'll be running gel electrophoresis, agar gel electrophoresis with 1% of gel, agar gel. So after that, I'll be observing the bands in the gel top. So I'll be posting that image in the end of the video. If you like it, you just do the experiment in your laboratory. If you found the result good, just make your comments in the comment section. Thanks a lot. Bye bye.